Welcome to Introductory Biology. In this first lecture, we will talk about the process of science as well as some characteristics of living things. We will talk specifically about the properties of life as well as different levels of organization within biological systems. This is called the biological hierarchy. We'll talk about the way science is done. We can contrast descriptive and experimental science. We can talk about induction and deduction. You will have a class handout about this as well. We will talk about the scientific method, and we will discuss whether or not it's possible to prove hypotheses are true. And spoiler alert, the answer is no, we cannot prove hypotheses are true. Let's start by going over the properties of life. Your reading lists several properties of life, including most of those listed here. I'm simplifying the list a little bit for the ones you are responsible for. We are also adding one, it's the first one, namely cellular organization. So your reading talks about this elsewhere. It specifies that all life is cellular. Most living organisms are composed of only one cell. This is true for basically all bacteria, for example, and also some more complex organisms. However, other organisms, like most plants, fungi, and animals, are multicellular, in which case they are made up of one or more than one individual cell. A second property of life is that there is order and also complexity, so we'll call that ordered complexity. Life shows sensitivity, it shows growth and development, it shows the ability to reproduce, the ability to use energy, the ability to maintain constant conditions within the organism. This is referred to as homeostasis, and across generations we can see evolutionary adaptation. Let's talk about those in some more depth. So cellular organization we just discussed. We said that life shows order despite its complexity. So these are two different ideas. Something that's complex has different parts. It is not homogeneous. If that entity shows order, that means that the different parts have some arrangement that is not random. So there's not a random hodgepodge of parts, but instead there's usually a pattern, and that pattern is typically associated with some function. This is a rock. A rock is complex. It has many different kinds of minerals, and you can see that with the different colors in this photograph here. However, it is not highly ordered. So these different minerals are more or less randomly distributed within the rock. Oops, that is not going to be true of living things. Let's get back to where we were. Okay, the third item was sensitivity. This means that organisms can perceive their environment and they can respond to the signals that they receive. Here's an example. This is compass plant. It's a plant that grows on prairies in the Midwest. And it is able to orient its leaves in an east-west direction. So one faces east and one faces west. This has the effect of reducing sun exposure. Since they're out on the prairie, they tend to get more sun than they really need. And by reducing exposure, it helps them to save water. And so in this case, what they're sensing is the compass directions, and then the response is orienting the leaves accordingly. Organisms show growth and development. As I discussed on the reading handout, growth is enlarging in size, while development is a little bit different. Let's shrink this down so we can see the words. Development, we'll say, is the formation of different structures that are associated with different functions. And so that is not just getting larger, but it's having parts that become more specialized. And here's an example. This plant here is a liverwort. It shows both growth and differentiation. And so growth would just refer to one of these lobes going from being small to being big. There's not really specialization on that lobe beyond just getting larger. However, an example of development here is that on some of the lobes, specialized organs develop, like shown in this blue circle, and these organs make asexual reproductive structures. So propagules that are able to go off and form new colonies of the plant. And so that specialization of parts is an example of development. So we just alluded to the fact that the liverwort can make propagules that are used to make new plants. That is an example of reproduction. And all living things need to be able to reproduce, or at least some members of their populations do, otherwise that population would go extinct. And so these asexual propagules, asexual means without sexual reproduction, are one example of how a plant can do that. However, liverworts as well as other plants can also do sexual reproduction, in which they make either eggs or sperm. Fertilization occurs, leading eventually to the production of a zygote, and the zygote eventually will form a structure that is a new generation of the plant. And so reproduction is not always sexual. It can also be asexual. The thing in common is that in both cases there's a way that the individual can reproduce and make members of the next generation. So we're up through 5 different properties so far. The next property we will discuss is energy utilization. Remember that living things cannot create energy. Animals and fungi and bacteria do not do so. Plants also do not do so. Instead, living things acquire energy and they can change its form. They can change it from being stored as one kind of chemical to being stored as a different kind of chemical, or they can release the energy, for example, through movement. So they use energy, they do not create it. And all living things, in fact, must use energy. So for plants, we know they acquire energy from sunlight through the process of photosynthesis. But that's not always true. So what we're looking at here is a parasitic plant, and you can see it is white, not green. So it's not green because it doesn't need chlorophyll, because it is not acquiring its energy through photosynthesis. Instead, it acquires its energy by parasitizing fungi that grow in the soil. Those fungi are probably attached to nearby plants, and so probably indirectly this fungus, or this parasitic plant, is actually getting its energy from nearby neighboring plants. The next process, or the next characteristic of life that we will discuss, is homeostasis. Let's take this word homeostasis apart into its 2 parts. The first part is the root homo, and homo here means like or similar. For example, a homosexual is somebody who is attracted to somebody of their like sex or their similar sex. The second part of the word is stasis. Stasis means staying the same. So if an organization is in stasis, it's not changing. So when we put these together, we see that homeostasis means maintenance of constant internal, let's change this to conditions, not characteristics. And this means that an organism can maintain a similar constant internal set of conditions even if the environment changes. One example of this would be a plant which is able to maintain the amount of moisture in its leaves. It does this partly by opening and closing openings on the leaves called stomata. So here is one stoma, and it's in the closed position. This is going to prevent water from evaporating out, so the water is maintained in the leaf. If the leaf has plenty of water, it can open those stomas, or stomata, and let water out. And so by this difference, the plant is able to regulate the amount of water and keep a relatively constant internal amount of water, regardless of whether it is moist and humid outside or whether it is dry outside and has not rained for a while. For humans, given that it's cold out right now, we can think about homeostasis as applying to our body temperatures. We stay very close to 98.6 degrees whether we are in temperatures outside that are 100 or 105 degrees, or if we're in temperatures outside that are at or below freezing, our body has mechanisms to maintain our constant temperature. Number 8 is evolution, and evolution refers to descent with modification. Now, in some ways, this property doesn't really apply to individuals. Instead, it applies to the difference between individuals and their offspring. I'm just going to resize this window so it's not behind where my image shows up on this slide. Okay, so we said evolution is descent with modification. Evolution occurs because individuals are typically not genetically identical to their parents. This is especially true when the individuals are made from sexual reproduction, although it also applies to some degree to individuals that result from asexual reproduction. Either way, if there is variation in offspring from a set of parents, then some of those offspring are going to have changes that better allow them to survive or reproduce. And if that's the case, then on average, those offspring are likely to leave behind more offspring than their siblings who have slightly less ideal traits. What this means is that over time, beneficial changes will tend to accumulate, especially if the differences in the offspring are heritable. So if they are in the DNA, for example. So evolution is really a process that happens between generations. However, we can see the results of evolution when you look at the traits or adaptations of individuals. So we could look at something like the stomata on leaves, and we can recognize that to be a trait that's favorable that resulted from evolution. We can look at what happens over time between generations if we want to witness the process of evolution occurring. So those were the 8 properties of life that you are responsible for knowing. As I mentioned, your book does break them up a little bit differently, but if you look at your reading handout, they'll be listed similarly to how I've listed them here. We are going to move on now and talk about the biological hierarchy. This is a general idea about levels of organization within living things. And we're going to start with the smallest levels, getting down to specific atoms and how they interact and form macromolecules. Macro means large, so macromolecules are simply large molecules. We then will think about how macromolecules can work together to serve specific functions within a cell. And these small units within a cell that have a specific function are called organelles. We'll see that all living things have organelles, although eukaryotes have a lot more, and they are more complex than do bacteria. We'll then talk about cells. I put an asterisk here because cells are the smallest unit of living thing that we consider to be alive in themselves. And so organelles are important parts of cells, but organelles are not themselves alive. But once we have a cell, an individual cell can be considered to be alive. We have in multicellular organisms that are complex, we often have cells that work together and are similar and serve a similar function. These are called tissues. Then we can have groups of tissues. So different kinds of tissues come together to serve some system, and that can give us an organ. So, for example, our stomachs or our eyes are organs. And then we can have organs that come together and function for some larger, or integrate together in some larger way for some specific function. So, for example, our eyes and our ears and our brains can all be considered part of our nervous system because they together allow us to collect and process sensory information. The next level of organization is the organism as a whole, and I think that is fairly straightforward. We then have levels of organization above the organism, so groups of individuals that somehow interact with one another. Our first level here is the population. And for all of these levels, population and above, we can see them on another slide, which I will bring up now, so they aren't jammed in so much. And so let's get back to what a population is. This refers to all individuals of the same species in a particular area. So a population could be all of the human individuals living within Spartanburg. It could be all of the fleas residing on a particular squirrel, or it could be all of the wildflowers of some species in one section of forest. If we think of all of the individuals of the same type that occur anywhere in the world, that is a species. So a species is really all of the populations of a given kind of organism. Above the level of species, we have communities, and a community refers to individuals of all different species that occur within the same area. So we could think of all of the different tree and plant species within the forest as being a community, or even all of the animals, fungi, plants, and microorganisms in the forest as a community. The next higher level is the ecosystem. This refers to all individuals that have the potential to interact. So as we just said, all of the microorganisms, plants, fungi, and animals in a particular section of forest, along with the abiotic environment. Let's take apart the word abiotic. Biotic refers to living things. The prefix a or an always means without. So the abiotic environment are things that are not alive within the environment. This could include things like the gases in the air. It could include things like the temperature. It could include minerals in the soil. And so when we start thinking about how all of those interact together, then we are thinking about the entire ecosystem. And finally, the biosphere as a whole is going to be the section of Earth within which all living things reside. So it goes just a little way into the crust of the Earth, and then a little way into the air because there are birds and microorganisms that float around. Anything within that envelope is considered to be inside the biosphere. That concludes the biological hierarchy. While we're talking about this, we should talk about why do we study all of these different levels of organization? Why don't we just study species, for example, or maybe study tissues or organs? The reason is the idea of the emergent properties. What this refers to is the fact that attributes of higher levels of organization are not obvious from the attributes of lower levels of organization. So if we go back to our list of levels, we might think that we could understand everything about organelles by studying the characteristics and properties of macromolecules. However, this doesn't really work, because to understand an organelle as a whole, we need to know both about the properties of the macromolecules that compose it, as well as how those macromolecules interact with each other. Similarly, if we want to understand how organs work, we have to understand the tissues that make them up, but we also have to understand how the different tissues in that organ interact with one another to form the organ as a whole. And so because these properties are not obvious, we need to study multiple levels of organization. Let's talk more generally about what we do in the process of science. And we are going to focus specifically on natural science. We'll define this as the practice of learning about the natural world by making systematic observations or testing hypotheses. And hypothesis testing is an important part of this. Many definitions focus on this to the exclusion of just making observations. I will argue that both are important. Either way, if we are studying natural science, we will rely on the rule called uniformitarianism. And this looks like a made-up word because it is so long, but if we take it apart, basically one part of the word is uniform. Uniform means same, and then the rest of the word just tells us that it is a belief system. So when we do science, we are following a belief system that says everything is uniform. What do we mean by that? Basically, it says that we believe that the fundamental laws of nature are constant across space and across time. So a hackneyed example of this, but one that is pretty common, is that if I were to drop a ball, then let's assume the ball is going to fall towards the ground. If I were to drop the ball again somewhere else, maybe in Africa or on a boat in the middle of the Pacific Ocean, I would still expect the ball to fall to the ground. That is because we assume that the law of gravity applies everywhere equally. Of course, gravity won't be as strong on a different planet or in space, but the fundamental idea that objects will attract each other and the strength of that attraction will be based on the mass of the interacting objects, that is going to be true anywhere in the universe, not just on the surface of the Earth. And similarly, we'd expect this ball dropping to work not just now, but also to work tomorrow morning, next Thursday, and in 1,000 years. So there won't be any change in that fundamental law of gravity across either space or time. If uniformitarianism isn't true, then we're stuck, because it means that when we make observations, those observations might not be similar to observations we would make at different places or at different times. Similarly, if we were to test hypotheses and find support for one but not another, if uniformitarianism was incorrect, we wouldn't be able to take the rule we learned from our hypothesis testing and assume that it would also apply in other situations. So this is really fundamental. It's interesting to think about. We can't prove that uniformitarianism is true. All that we know is that it has worked for us for a long time, and we know that it's extremely useful. So philosophers like Hume argue that based on that premise, we should go forward with its use, even though we can't, from first principles, explain why those rules are constant over time. We talked a little bit already about emergent properties. We said that they were attributes of higher levels that were not obvious from lower levels. Let's go back to that idea just for a minute and look at a couple of examples. And so one example would be in physics. We can understand properties of individual molecules. But if we want to understand hurricanes, we also need to think about how those molecules in the air would interact with each other under different levels of heat in order to create wind currents. If we're thinking about macromolecules, then we're going to think about the properties of the individual atoms that make them up. But we also have to think about how those individual atoms lead to chemical properties that are going to cause our long protein molecule to fold into some particular shape, which will then determine its function. At higher levels of organization, we could think about something like a population. So a population, remember, is a group of organisms of the same species in an area. As soon as we have populations, we can start thinking about things like density. How many are there per unit area? We can think about their dispersion. Are they all spread out equally, or do they occur in clumps, with one clump maybe in one region, and then an area with few individuals, and then some other area with a higher density again? We could think about the sex ratio. Are there half male and half female, or are there more females, or are there more males? And we could think about age distributions and other aspects of the population. So these are all things that wouldn't make sense to talk about if we're just focused on an individual. These properties instead emerge once we have multiple individuals interacting together. As a final example, we could think about things in behavior. So we can study the behavior of individual fish, but that wouldn't tell us much about swarming behavior, like this schooling behavior we're seeing in fish, where all of these fish swim off in the same direction. To understand that, we have to think about how the fish interact. So those are all examples of emergent properties. Emergent properties, then, tell us that higher levels of organization are always more complex than the levels beneath them. This complexity poses a challenge. It's hard to understand complex systems. And so, sort of paradoxically, what we do in the process of biology is we study the more complex levels by first studying the lower levels and then putting that knowledge together. This general approach is referred to as reductionism. And so that's a pretty simple word. Reduce or reduction makes smaller, and ism, it's a belief or a practice. So in this approach, we understand a complex problem by reducing it to a study of its individual parts. Let's think about what that looks like. And so you've probably guessed by now that I'm a botanist. And so we'll look at a plant example here. And in this example, there are these weird flowers that occur in members of this genus, Banisteriopsis. And in this group, I won't even try to say that word a second time, by the way. In this group, there are these weird glands on the sepals that I'm showing with the mouse right now. So they occur near the base of flowers, and it's of interest to biologists. Why are they there? What function do they serve? And so how could we go about answering that with a reductionist approach? Well, according to reductionism, we don't want to try to consider everything at once. Instead, what we would like to do is break this down into a series of smaller questions that we can answer, and then put all of that knowledge together. For example, one question we could ask is, what is the chemical composition of the products made in these glands? It turns out that it's some particular mix of carbohydrates, fats, and proteins. We could then study each of those to understand where are they found in other organisms and what are their typical functions in other organisms. That would be one way we could reduce this problem. A second thing we could do is we could observe the plant and see how do animal visitors interact with these glands. Do they stay away from them? Are they deterrents? Or are they attracted to them? Do they gather one or more of those substances from them? And so a behavioral approach would be a second approach that we could use to look at this. We could also use an experimental approach where we remove the glands and we see what happens to the flowers compared to other flowers for which we leave the glands in place. If the flowers are successfully pollinated with the glands and not successfully pollinated without, that tells us something then about the fact that the glands are used to attract pollinators. If beetles eat the flowers without glands, but they leave flowers with the glands alone, that might tell us the glands are there to make something that deters herbivores. Once we do all of these different approaches to looking at what the glands do, then we can put that knowledge together to come up with some coherent idea about their function. And by the way, their function does seem to relate to attracting pollinators. Let's talk a little bit about descriptive and experimental science. These are two legitimate approaches to asking scientific questions. In descriptive science, we are trying to document patterns, but we are not testing hypotheses. The description might lead eventually to a hypothesis, but it doesn't start with it. And I gave you some examples of this, both in your reading and on the reading handout. So for example, if we went to a comet or the moon and we collected minerals to see what minerals are there, we would be doing description, but it's still science. Another example would be if we sequence a gene for which we don't have any prior idea of what the sequence should be, we're gathering information that relates to biological systems, but we're not testing a hypothesis. Again, that is descriptive science. And if we make a list of all of the plants in South Carolina, that is also descriptive science. In contrast, we have hypothesis-based science. In this, we attempt to support or disprove proposed explanations. Our hypothesis is a tentative explanation for a phenomenon. And here we are explicitly doing experimental science because we are going to typically do some modification to see whether our tentative hypothesis is supported or whether it is refuted. So an example of this would be with the plant example. If we removed these glands and we observed how pollinators interact with the flowers with and without the glands, we might have the hypothesis that the glands attract pollinators, and we can test it with that experiment. That is then performing a manipulation and doing hypothesis-based science. Let's talk about the terms hypothesis and theory. A hypothesis, as I just said, is a tentative explanation for an observed phenomenon. It is not necessarily the correct explanation, and we might even have multiple alternative possibilities or alternative hypotheses for something that we observe. For a hypothesis to be useful, it should be reasonable, so it should be something that we think might be the correct answer. It must be testable, so there has to be some way that we could potentially falsify it, otherwise it is not useful. And it needs to be based on the laws of nature. So it should be based on something that we think is uniform and would be repeatable in other places and at other times. Our hypothesis is tentative because we might find evidence that suggests it's completely wrong or at least partially wrong and needs to be modified. So we never totally accept it. We always allow the possibility that we could eventually find evidence that makes us reject it. So I just resized the window here to make sure we can see the text. Let's contrast the hypothesis with a theory. A theory is a tested and confirmed explanation for observed phenomena. Usually our theory is going to have multiple supporting lines of evidence. So in other words, we have not just tested it once, but we've tested it repeatedly. A theory like a hypothesis is still tentative. Even though it's been tested and confirmed, we will leave the possibility that we will eventually find evidence that disproves the theory. For example, Newton's laws of motion were widely accepted as being true, and then later with Einstein, we learned that in fact they are simplifications that don't apply in all situations. And so we modified his theories to reflect those limitations. We will move on here to talk about two processes we use to learn about the world. And these processes are induction and deduction. This is one of the ideas that is often challenging for students, and so we'll spend a little bit of time on it, and hopefully we will have opportunity to work on some examples in class. Let's start with deduction. Deduction is the use of logic or mathematics. There's some equivalence between these, so mathematics is based on the rules of logic. So continuing, it's the use of logic or mathematics to consider a problem or to make predictions. The nice thing about deduction is it should necessarily give us correct answers as long as we don't make a logical mistake. In contrast, induction is the use of observations to arrive at a general principle. So we might observe several things, think about their similarities, and then make some statement about what those similarities are. Hopefully this will also lead to correct knowledge, but there's more room here for us to make observations that in fact don't lead to a correct conclusion. Let's say a little bit more about deduction. We've already defined it. Deduction could be based purely on ideas without any evidence. So for example, let's consider this, this scenario. If a coin has a head side and a tail side, most coins do, when this is the case, we know that if the head side is showing, then the side with the tails on it must be facing downwards. If we were in class, I would do this example, but I think you can sort of imagine it and understand why that is true. Conversely, if the tail side is showing, we can conclude that the head side must be facing downwards. And so this is logically necessary for a coin with a heads and tails side. And therefore, there's not really the possibility of us being wrong. If the premise is true, here the premise is that the coin has both the head side and the tail side, then our conclusion must be right, that if one is showing, the other one is not. In practice, when we do deduction in biology or in science, often what we're doing is we are saying there's some general pattern that describes a characteristic of a group. And we then say, if some specific individual is a member of that larger group, then it should also have that characteristic. I will go over an example of that I think will be more clear. So here's an example. I'm going to get rid of this. This was from our coin example. Let's use the example, all peaches have a pit. The pit is that hard part that you can't bite through in the middle. If this fruit is a peach, it also should have a pit. So let's look at this statement. What is our premise here? The premise is that all peaches have a pit. If that's correct, we also have a second premise. Our second premise is that the fruit that we're looking at is a peach. So if this is correct, and it's correct that we're looking at a peach, it must logically follow that that peach will have a pit. There's not really another possibility. If we were to bite through the middle of it, and in fact there was no pit, then we would know that the fruit we were eating was not, in fact, a peach. That conclusion would not be a mistake about deduction. Rather, it would tell us that one of our premises must be wrong. Either all peaches do not, in fact, have a pit, or the fruit itself is not a peach. Those are the only two possibilities. We will now look at an example of induction. Induction is usually about describing patterns, the use of observations to arrive at a more general principle. So here's our example. Let's imagine I measure the size of 100 male flowers and 100 female flowers for some particular species. And I find that the male flowers are all larger than the female flowers. From this, I can come to a general conclusion that for this species, male flowers are in fact larger than female flowers. That's not necessarily true, because I did not measure all male flowers of this species, and I did not measure all female flowers. I just measured 100 that I found, in other words, a sample of the male ones and a sample of the female ones. It might be that the 101st male flower is actually quite small, or the 101st female flower is actually quite large. So with induction, there's always the possibility that our rule is not correct, unless we have literally observed every single individual. And that is virtually never the case. Our next step here is to talk about the scientific method. And I know from state standards that you should have had the scientific method several times between first grade and wherever you are now in school. That said, it's a super important idea, and it is really instrumental in the development of science, and so we will go over this. The scientific method always starts with some observation that leads to a question. So there's something we want to know. Typically, we do some background research. We Google it to see if somebody already knows the answer, or we go to the coffee maker and we ask our colleague if they know, or maybe we look it up in the textbook. Let's say that we can't find evidence that somebody already knows the answer. The next thing we do is we construct a hypothesis. In other words, we come up with one or more than one potential explanations that are testable and might explain whatever it is that we saw. Next, according to the scientific method, we will do a test. And ideally, the test will let us see whether one or more of our explanations is consistent with the experiment that we perform. Hopefully, some of them are ruled out, and in the best of all worlds, only one of them remains that is not ruled out. If that is true, then we can say, we do our experiment, we analyze the results, and from that we draw a conclusion. There are ideally two conclusions we can draw. One is that the hypothesis, the tentative explanation, is not consistent with the experiment we performed. In this case, we can say that our tentative explanation is wrong. It might be completely wrong, or it might be partially correct, but at least partially wrong. So either way, we need to change that hypothesis a little bit or a lot, come up with a new hypothesis, and then retest it. Another possibility is that the results we get are consistent with our hypothesis. In other words, they're what we'd expect if our hypothesis was correct. In this case, we're going to say our hypothesis is tentatively supported. In your book, they say the hypothesis is correct. That statement is not, in fact, correct. We can never say that the hypothesis is correct. We'll only talk about levels of support. So it might be supported strongly, it might be supported weakly, but it has some tentative level of support. Whichever result we get, we should tell other people about it. That might mean going to the coffee maker and talking to our friends or colleagues. It might mean writing our results up in a scientific paper and sharing them with the world. It might mean writing something about it on the internet. But there's some sharing that goes on. Now, this isn't in your book, but it should be. Even if our hypothesis is supported, we can't conclude that we were correct. Instead, we need to think of a new way of testing our hypothesis, or think of some related hypothesis that goes a little bit further, a slightly more complete explanation, and then think about how we can retest it. So usually the process doesn't stop. Instead, it is iterative. Whichever way we go, whether we were wrong or supported, we go back and we think about new hypotheses. Ideally, when we do the scientific method, we don't just test one hypothesis at a time, because if our hypothesis is disproved, then we don't really have much that we've gained. If we can test multiple hypotheses at once, then we hope that all except one are rejected or disproved, and there's only one left. In that case, we have a high level of tentative support for the remaining hypothesis. So let's ask, can the hypothesis be proven true? And let's answer that question. And let's answer that question in big font. Let's go up to 89 font, and maybe we can even put it in red. The answer to this, oh man, that's not 89 font. The answer to this is no. We can never prove a hypothesis true. And instead of explaining why, I think it is more productive and effective to show you an example. So let's start with a hypothesis that we all know is not correct. The hypothesis is that flies can form spontaneously. In other words, not from eggs. They just form out of the flesh of meat. And they do this if they are in the presence of fresh air, like this meat over here that's exposed to air. but that they cannot do it if they are in stagnant air. So if we put this glass cover over the meat, the air becomes stagnant. It can't move around very much. We are hypothesizing that under these conditions, we cannot have spontaneous formation of flies from meat, but under these conditions we can. So is this testable? Yes. Is it a tentative explanation? Yes. Is it consistent with what we know? Well, we know that when meat is exposed to air, eventually flies start to form. And we know that when meat is not exposed to air, flies do not form. So yes, it's consistent with things that we know. It's a reasonable hypothesis. So let's say we actually did this experiment. We expose this to air, we don't expose this to air, and then we see whether there are maggots forming in the two pieces of meat. Well, we find that there are, as we predicted, there are magnets in this piece of meat, but as we predicted, there are no magnets in this piece of meat. So the results are consistent with our hypothesis. Does this prove that our hypothesis was correct? No, it does not. The reason it doesn't is that there are other hypotheses that could make the same prediction. For example, an alternative hypothesis is that maggots only form in meat when female flies lay eggs into that meat. Therefore, if we expose meat to female flies, then eggs can be laid and maggots can be formed. But if we prevent flies from accessing that meat, then eggs cannot be laid and maggots will not be formed. So we have two different hypotheses that make the same predictions. Even though our results were consistent with our first hypothesis, it doesn't show that the first hypothesis was correct because our second hypothesis also made the same prediction. The lesson from this, and this is important, and I cannot promise it will be on an exam, but I can promise it has been on past exams. The lesson is that hypotheses may be supported, but they are not proven true. This is because different hypotheses can make the same prediction. So that comes to the end of our discussion of the first chapter of the book. I want to remind you about the structure of this course. This is not a lecture-based course. Instead, the primary way you will learn the material is from your readings, and your learning will be organized on the reading handout. So your reading handout is really the guide to what you need to know, and the reading is where you get that information. The lecture is a supplement to help reinforce the ideas. So this is a long way of me saying that you need to know everything in today's lecture, but if there are things on your reading handout that we didn't discuss today, you should still know them. Use the reading handout as your primary guide when you are studying. To summarize the ideas that we talked about in this lecture, make sure that you can recall and define the different properties of life, the 8 that we discussed. Make sure you understand the difference between induction and deduction, and if I gave you an example of a process, make sure you can identify it as one or the other. Be able to explain what reductionism is and why it's useful, but also be able to explain why emergent properties mean that we can't only study lower levels of organization to understand higher levels. Know the different steps in the scientific method, know that we cannot prove hypotheses to be true, and know that the method is iterative. It keeps going back to new hypotheses and new tests. And I've already said that, so I will not belabor the point.